see. Oh, am I allowed to share, Pat?
Yes.
My and it's grayed out for me. I'm not sure if the settings are.
Let me, does that help?
Let's see. Yes, it does. All right. Everybody see that okay?
Yes.
Okay. Well, I told Pat earlier, but if you have any questions as I'm going through this, just don't hesitate to stop me and ask. I'll try to make this as clear as possible, although it can get a little wonky sometimes. So today I wanted to talk to you a little bit about some research I did back when I lived in Texas. And the reason I wanted to do that is because I figure y'all might get a little bit tired of hearing about the stuff from around here that we're all researching. And so I wanted to introduce you to a system you may be less familiar with, which is some of the aquatic salamanders down in the Edwards Plateau region of Texas. And so just to get us started here. So just so you know my research interests, I'm a behavioral evolutionary ecologist. I've worked with both fish and amphibians. I'm especially interested in predation and trophic relationships, and especially the intersection of sort of human-caused change and how that influences evolved ecological relationships that we see in organisms. The other thing I want to point out is I am a behavioral ecologist, and one area that has been lacking in the realm of conservation research for a long time is the role of behavior in that research. And so if we think about the fact that we have anthropogenic impacts on animal behavior, so we change things and it influences how animals behave, um, what we've been really good at is finding those behavioral indicators. But what we haven't been really good at is applying a behavior-based management strategy to address any issues that we cause. This is from a review paper that was published in 2016 where they really highlighted the role for behavior in conservation biology. And if you look at conservation behavior papers, this is a heat map. And so the heat map shows the redder it gets, the more there are, is basically the way to read that. And we see that there's been a lot of research on dispersal and migration, as well as foraging behaviors. But a lot of other fields of research are just horribly underrepresented in conservation behavior work, such as anti-predatory behavior, which is what we're going to be looking at today. And if we look at behavior specifically as a subset of all conservation biology papers, we find that behavior is sorely underrepresented overall, with the few exceptions maybe being looks at dispersal and migration. So for example, we now have implemented migration corridors to help connect what were disconnected populations due to things like, say, roadways. And so when we're thinking about predator-prey relationships, one of the reasons that I focus on predator-prey relationships is because fundamentally when we think about the things that influence biological fitness, which is a measure of reproductive success, fundamentally it is the foundation of whether a population survives or not. We can sort of categorize all behaviors into three categories that influence it. We have reproductive behavior, we have foraging behavior, and we have what we can think of as survival strategies. And if you were going to put avoidance of predators under one of these three categories, where would you intuitively put it?
Survival?
Yeah, right? Obviously, predators eat you. You don't want to be eaten if you're a prey. And so you'd throw that up under survival strategies. But the more we research the influence of predators on prey, the more we realize that the presence of predators, just living in the presence of predators, actually influences reproductive behavior and foraging behavior as well. And this realm of research, these indirect effects of living under the risk of predation, are what we call the ecology of fear. And so we were just chatting, a couple of us before we started, about how the presence of wolves in Yellowstone influenced how elk used the habitat, which had rippling effects throughout Yellowstone National Park. That all falls into the ecology of fear research, right? The pure presence of the wolves changed the behavior of the elk, which changed their overall biological fitness. The reason I focus on freshwater systems primarily is that freshwater ecosystems, while they're only 1% of Earth's total surface area, they contain 10% of known species, known aquatic species anyway, and a third of all vertebrate aquatic species. And so they disproportionately represent a huge amount of biological diversity. And so they're really important to study and understand. And the reason I like to focus on amphibians is because for about 40 years now, we've been documenting a global amphibian decline. We estimate that at least 30%, if not more, of amphibian species throughout the world, so frogs, toads, salamanders, etc., are either in decline or likely extinct. This is not concentrated in just, say, tropical rainforests. This is a global phenomenon. And so I feel that we need to study it to understand it better. Also, amphibians, because they often have both aquatic and terrestrial life stages, they represent a really important interface between land and water. So they transfer nutrients, etc., between the two. And so today, here's what I'm going to talk about. I'm going to talk about some of my research specifically. We're going to get into the nitty-gritty of predator recognition and avoidance and how this plays into the ways we think about conservation and how we interact with these species. So first, to give you some background on recognizing and avoiding predators. So when we think about predator recognition and avoidance, it's really about just staying alive in a dangerous world, right? Predator avoidance is fundamental to survival. And effective avoidance really involves just three basic stages, right? You have to detect a predatory stimulus. You have to recognize that stimulus as being threatening. And then you have to employ an avoidance strategy of some sort. And so if we look at this picture, which by the way isn't real, it's photoshopped, but if we look at this picture of a lion and zebra, right, we can ask ourselves, is this an adaptive predator avoidance strategy for this zebra? And in most situations, we would say probably not, right? But I could show you a few ways why it might be, and that's what we're going to get into. All right. So first and foremost, predator detection and recognition. So detection is just detecting a predatory stimulus. That stimulus could be a visual cue. They see the predator, right? They could smell the predator through olfactory or chemical cues. They could hear the predator through auditory stimulus. When we're working with amphibians, it's usually going to be a chemical cue detection. So they're either smelling it or detecting it in the water. We call those chemical cues. And I showed a picture of a salamander over here doing what's called buccal pumping. And buccal pumping can do, it has two purposes. So one, it can help them pull in oxygenated water, which they then can extract the oxygen from the water, but it also helps them pull in any molecules that would constitute chemical odors in the water and better smell those, better read those. And so recognition then is not only did you detect that stimulus, but you recognized it as a threat. And that recognition, right, it can be innate. So if you have a long coevolutionary history between a predator and a prey, often those prey are born with the ability to recognize that particular predator stimulus as a threat. A lot of prey, however, can learn to recognize new predatory stimuli. And also many prey will exhibit what we call neophobic responses. And so it may not be that they recognize a particular stimulus, but if it's a new stimulus that is unrecognized but detected, they'll still respond with a fright response. And so that would be neophobic. One thing that is important to recognize is that the ability to detect a particular stimulus, that's going to be genetically predetermined. Either you've got it or you don't. But recognizing, associating that stimulus with a threat and or responding in an appropriate way, that's plastic. That can be altered with experience of the individual through life.
A question?
Sure.
What does plastic mean versus elastic?
Versus static. Yeah,
I
yeah.
think
No, good question. Thank you for asking that. Yeah, plastic would just mean that it's you can change it. All right. So our third step, predator avoidance. Predator avoidance is basically prey have two categories of option. You can either avoid detection, right? So you detect a predator, what do? Well, you might, if you're moving around, you might reduce activity, right? So they may be more likely to see you if you are moving. Take cover in refuges, under rocks, under logs, whatever the case may be. You could shift daily activity patterns or locations. So maybe if your predator tends to be more active during the middle of the day, maybe you become more active at dawn and dusk or overnight, things like that. Alternatively, if you can't avoid detection, you have to avoid capture, and that's where you get into your fight or flight responses. What's the best to do? Now, you'll notice the picture I put on here underneath. Those are our little salamanders. They're all hiding. This is what they look like in captivity during the day. They all hang out together underneath these artificial refuges that we give them. But all right. So thinking about evolutionary relationships and predator recognition of native and novel predators, I'm going to introduce here our study system and we'll talk about it a little bit. So this is the San Marcos salamander, Eurythia nana. It's a species that is endemic to and found only in the headwaters of the San Marcos River, which is called Spring Lake. So they only occur there and up to about 50 meters downstream, and that's it. Well, the San Marcos River, the headwaters are in the town of San Marcos, hence the name. And originally, this was an aquifer-fed kind of flowing swamp. But people came in, dammed it up, and so now the headwaters is a flowing lake. It's still aquifer-fed, crystal clear, it's beautiful. All right. Our salamanders, they're totally aquatic. They never take on a terrestrial stage. It's what we call pedomorphic. So they retain that aquatic larval stage through their whole life. You can see there that picture I took is a female up against the aquarium and all those little white things in her belly. Those are her eggs. She's gravid. The reason I wanted to study the San Marcos salamander is partly because the proximity made them convenient to study when I was interested in these questions, but also because this particular species is part of a larger salamander complex. The San Antonio-Austin corridor, I-35 corridor, sits on top of an aquifer system that feeds a lot of springs and rivers throughout the region. And within this system, there has been immense diversification of salamander species, many of which are found only in one or two springs, or sometimes they're cave adapted to live down in the aquifers, like the Texas blind salamander. And so these are a really unique species set. And sadly, there'd been a few researchers working hard on their genetic relationships to understand the evolutionary relationships between them, but we knew virtually nothing about their ecology, including how they were interacting with their predators. And so I was the first one to sort of dig into that with these species. Just to show you how many of these species were threatened or endangered, this is a publication. It shows you sort of the distribution of some of these species or species complexes. Each one that I marked with a red arrow is a species that is listed under the Endangered Species Act as federally threatened or endangered. All right. So our San Marcos salamander, I said it's in Spring Lake. It coexists there with a lot of fish predators. And some of these fish predators are native or to the habitat, and others have been introduced via sport fishing. So a lot of us, for example, are familiar with a red-breast sunfish. They're a native species here to the Northeast, right? But naturally, they don't occur in the Midwest or in the West. but they've been introduced here in Spring Lake and have done very well because of the nice cool aquifer temperatures of the water, and are one of the primary predators on this salamander. So just to give you an idea of the type of, this was some underwater footage I took. It'll clear up, I promise. But I just wanted to show you this so that you had an idea of the sheer abundance of predatory fish residing in this habitat. The salamanders are little tiny salamanders, and they live underneath all of these rocks along the substrate. And you'll notice these sunfish and bass just hover over the bottom, and they're just waiting for one salamander to make a mistake, and they scoop it right up. Also, if you know anything about sport fish, you'll recognize that many of these fish, they look emaciated. They're starving. The density is so high of these fish that they pose a really significant predatory threat to these salamanders.
Can I ask how big the salamanders are?
Yeah, they're about yay big, about an inch or two at most. Their nana means small or minute in Latin, and so the species name Euryssia nana indicates how small they are, dwarf. Yeah. So yeah, I won't make you watch that further, but just to give you an idea of what's going on with that. All right. So thinking about experience that salamanders have with predators, this species was also an ideal species to work with because not only could I collect them from the headwaters of Spring Lake pretty easily through snorkeling, I had access to a captive population that's maintained at the San Marcos Aquatic Resource Center, which is run by the U.S. Fish and Wildlife Service. They're the ones in charge of monitoring Endangered Species Act protections. And so because there were so many unique endemic species throughout the region, they maintained captive populations of them at the hatchery on the off chance that, say, there was a chemical spill at the headwaters of Spring Lake that wiped out the entire species, right? Well, then they would have some individuals to reintroduce. And so because they were breeding these individuals there, I had access to adults that had never had any experience with predators at all. And so that meant I could ask questions about how important experience with predators was in influencing their behavioral responses. So the first question I really wanted to get at with these species, because there are so many introduced sport fish species that pose a potential predatory threat, we knew from some other systems that some prey don't innately recognize the stimuli of non-native predators as threats. And so they don't respond to them in the same way as they do native predators. And so we were concerned that there were so many red-breast sunfish in here. Were they posing a threat to these salamanders that wasn't being mitigated through standard avoidance behaviors? And so I wanted to know two things. Do they recognize the red-breast sunfish as a predatory threat? And how does experience in the natural habitat affect those avoidance responses? So for example, even if the naive salamanders, predator naive salamanders from the hatchery don't recognize them, can they learn to recognize them by being in the natural habitat? So to address these questions, I compared avoidance responses of adults to the chemical cues of fish. I tested both predator naive fish, which were first generation captive reared. So they were from eggs that were laid in the hatchery and they'd never had any experience with predators. And then I also collected adults from the natural habitat. So they'd grown to adulthood under the risk of predation, under the threat of predation. The treatments I used in this first study, I used a native predator, the largemouth bass, a non-native predator, which is the red-breast sunfish. I used a native non-predator, the large spring mosquito fish, which is not to scale there. It's a little tiny mosquito fish. And then a sort of a second control, which was only water. So I had two versions of control, just blank water or a fish odor that should not be a predator. to get the stimulus, and I use these similar methods throughout the rest of the studies I'm going to talk about, so I wanted to make sure you understood them before we move forward. The stimulus is really just water that smelled like the fish. That's what the stimulus is. And so I would capture the fish from the San Marcos River headwaters, so they came from the same habitat. I housed them in 24 hours in an aerated tank. I released the fish, and then I collected the water they'd been in for the last 24 hours into individual aliquots. I froze it, and then I only thawed it right before I used it in testing. To test the responses of fish, or of salamanders to fish, we did it in the dark under red lighting. And the idea is that I needed to see the behavioral response of the salamanders to these predators. And the most common anti-predator behavior of salamanders is a reduction in activity. And so I did a pre-stimulus measure of activity. So for 10 minutes, I just recorded the amount of time that a salamander spent moving around an aquarium. And then I introduced one of the stimuli into the tank. I didn't know what treatment was going in, so I couldn't introduce my own bias in that. And then right after I introduced that stimulus, then I recorded activity again for the 10 minutes right after that. And so then for the response in this particular first study, I calculated it as a relative activity score. So I just took the post-stimulus time divided by the pre-stimulus time. And what that means is that if you get a number around 1, it means they didn't really change how active they were in response to the stimulus. But if you got a number bigger than 1, it means they were more active. And if you get a number less than 1, it means they reduced their activity level. And so first, looking at the results of the predator naive individuals, the salamanders that were reared in captivity, so they had no experience with predators. We found that they did reduce activity in response to predators, and that reduction was not different between the native and the non-native predators, the sunfish and the bass. And so that's an indication they do innately recognize the stimuli of both of these predators as a predatory threat. Where it got sort of interesting is when we compared responses between the experienced and the naive salamanders. So the experienced salamanders came out of the wild as adults. And what we found there is that we got weaker responses. There was still some response, but they were definitely weaker than the responses we detected in our captive reared salamanders. And so something was happening in the environment that was sort of softening up these avoidance behaviors that we were measuring. And so we thought maybe they're learning about risk, right? And so the take home from that first study is that because of the close evolutionary history, these species may have similar chemical cues. There are native green sunfish in that habitat. And so maybe the red-breast sunfish smells similar enough that they're able to generalize that recognition and response from the green sunfish to the red-breast sunfish. And so we thought we would test that, like how broad can they generalize if they're generalizing? And so in a follow-up study, we tested the responses of naive salamanders. So we used only those that were captive reared to the native green sunfish, the non-native redbreast sunfish, and then we also added in a non-native pumpkin seed sunfish that has not been introduced in that habitat. So it's not there at all. And then also to a non-native Rio Grande cichlid, which has been introduced into that habitat. And so sympatric means that they live together, they're in the same habitat. Allopatric means that they're in separate habitats. And so we wanted to see how broad this generalization went. And what we found is that the salamanders responded similarly to all of these predator odors. And so the thinking was that either they're generalizing across these groups, or maybe there's something else they're cueing on from the odor from these fish that's helping them respond similarly. This part told us, though, that maybe the introduced predators in this system, they might be less of a problem for these salamanders than they've proven to be for other species of amphibian where fish have been introduced. And so that took us to our next level. So thinking about risk assessment, what's going on in these relationships. So we know that predator avoidance can be really costly, and this is something that catches people by surprise. So we know that avoiding being eaten is definitely good, right? But the time that you spend, say, not moving around or hiding underneath your rock, it's also time that you're not out there foraging, you're not finding a mate, you're not doing these other things that do help you when it comes to biological fitness. And so the idea is that prey should respond to predators in the most efficient way possible. Enough of a response that they're likely to avoid predators, but not so much that they incur a whole bunch of opportunity cost. And so the benefit is staying alive, right? But there is the cost, an opportunity cost. And so if we think about how much time that prey individual hides, how long they spend hiding after they detect a predator, right? There's going to be a decreasing benefit from that because that predator is likely to move on or find another prey individual. And so you're still incurring cost while the benefit is not really increasing. And so the idea here is that we want to maximize the difference between the benefit and the cost. And that should predict how intensely prey should respond to their predators. Now, when we think about this and we consider the potential of a higher risk predator, so a predator that poses more of a risk than another predator, that benefit curve, that changes, right? If it's a more dangerous predator, you gain a better benefit from responding stronger to that predator. It shifts how intensely we should respond. This is, if any of you are familiar with general economics, a lot of ecology is just economics, but with a different payment method. In our world, it's usually energy or fitness. All right. So we want to minimize, a lot of prey have been shown to minimize opportunity costs by assessing the risk posed by a predator. How strongly should you respond? And one way that I was thinking that these salamanders may be assessing risk is maybe it has to do with what a predator is eating. And so it's less about the species specific odor itself and more about the part of the cue that comes from waste products from the diet. So can they tell that a particular fish has recently consumed salamanders or not? Maybe those pose the higher risk. And so what I did was I designed a study where I collected predators um, from the natural habitat, but I housed them for an entire week in flow-through raceways so that I could clear any diet cues, and I just fed them earthworms. And then right before I collected their stimuli, the 24 hours before, I fed half of those fish salamanders, and the other half I fed just earthworms. And so what I was able to get then are stimuli treatments. I used two different predator species, and I also had some that had not eaten salamanders for over a week, whereas some that had just recently consumed salamanders. So the cues might smell differently. And if we first look at the naive salamanders, so the salamanders from captivity, we get exactly what you might predict if salamanders are assessing risk. And that's that when you look at the responses to the fish stimuli from individuals that had only been fed earthworms, they still respond, but it's a weaker response than what you see when you compare the response to the stimuli of the same fish that had recently consumed salamanders. And so in other words, if there were degraded salamander cues in the waste products or the odor of those predators, salamanders were able to detect those and they responded more strongly. Where it got interesting though is, again, we pick up this sort of odd pattern when we tested predator experienced individuals. So salamanders that we'd collected from the wild as adults, they had experience with these fish. And so they showed a little bit of a response, but it was definitely weaker and statistically hard to detect. And so I ended up in this situation where, I've got the responses I get when I've had really no predator experience influence on these behaviors. And then I've got these weaker responses from salamanders that came from the natural habitat, so had experience with these predators throughout life. So the question became, what is going on in the natural habitat that causes these weakened responses? And so I hypothesized that it might be threat sensitive learning. And so experience with predators can affect the perception of risk. So in other words, are salamanders learning that these predators are actually less dangerous? So for example, if you detect their chemical odors again and again and again, but without that reinforcing connection of either salamanders in the diet, right, or maybe other chemical alarm cues, maybe you don't respond as strongly to them anymore. And this habituation to predator odor falls in a category of what we call adaptive forgetting, right? And so it's adaptive to stop responding to predators if they don't appear to pose a very significant threat, right? Again, you're minimizing that opportunity cost. Associative learning has been studied in a lot of ways, right? And especially in prey. And so associative learning in general is when you pair a known stimulus with an unknown stimulus. We all learned about this when we learned about Pavlov's dogs, right? You ring a bell, they salivate. The same thing can happen when you're learning to recognize a new predator odor. So one way that a lot of aquatic prey learn is that they learn to recognize, or they innately recognize the odors of injured conspecifics or damaged individuals of their same species as a threat. And if you pair that odor of injury, with an unknown odor, like say a fish you didn't recognize before, then later you recognize that fish odor alone as dangerous. And so that's how a lot of predator learning happens. And so if this is happening, right, I expect to see the similar thing in my salamanders. And so I had to do two things. One, I needed to know, do salamanders recognize alarm cues, these cues from damaged salamanders as a threat? And so I just did a really quick study. We were um, we were euthanizing salamanders for a different reason as another study. And so I was able to use their bodies first, right after they had been sacrificed, to collect damage cues by just scraping up their skin and running water over it. And that's what we use as the chemical alarm cue. And so when I recorded the changes in activity, right, 0 would indicate no change, negative numbers indicate a reduction in activity levels. What we found, just really straightforward, right, they reduced activity in response to water that was tainted with damaged skin extract from other salamanders. So they do recognize damage as danger. And so I said, okay, well, let's actually give these naive salamanders, these captive salamanders, very specific experiences with predator odor and alarm cues. Let's see what happens. Can I cause them to forget or habituate to predator odor when there's no reinforcing mechanism? And so over 10 days in captivity, I exposed the salamanders to 2 different frequency treatments, so either high frequency or low frequency, to either a predator odor plus alarm cue, so I call that the high risk, or just the predator odor only, so no reinforcing mechanism. Would these species habituate to predator odor, or would those individuals in that treatment habituate? And then I compared their anti-predator responses and sort of general activity levels before and after these 10 days. And so the results were a little bit wonky, but in general, kind of the 2 clear patterns that we found, right? We found that salamanders were less active after they'd been exposed repeatedly to predators. And so they were reducing activity levels when they were being continuously exposed. And they also responded more intensely to predator odor after repeated exposures. And so exactly the opposite of what I thought might be happening. In other words, I thought they might habituate to predator odor. Well, instead, they actually became more sensitive to it. They responded more strongly. And so this didn't give us a good explanation, at least in the short term, for what was going on to the predators. And so this was sort of the end of my research down there because I was moving on to the rest of to new research and some other less related stuff. But um, fundamentally, what we think happened. So this is my wrap up for you. Um, we had talked about how salamanders might respond more intensely to a more dangerous predator, and we did show that they were doing that when it was based on diet cues. Well, the other part of that that we didn't really talk about was the cost curve. Salamanders in the natural habitat, they're living under that constant risk of predation, right? Which means that they're not foraging as much and they're not mating as much, and their overall fitness is being affected in general. Whereas in captivity, they were fed as much as they wanted to eat. They were never under risk of predation. And so if we think about that, the opportunity cost for a wild salamander is probably a lot higher as far as avoiding predators than it is for a captive salamander. Captive salamanders got fat reserves out the wazoo. Wild salamander, smaller, hasn't had time to build those reserves. You take advantage of the time you have to eat. And so if we think about that as far as this goes, right, maybe our cost curve shifts a little bit. And what that means is that um, it could also mean that our wild salamanders, just to reduce those opportunity costs, really should respond less intensely to predators than our captive salamanders were. There were ethical issues when working with a federally protected species as far as starving them. And so we didn't do that in captivity, but that would be the ideal study to see if you got them into poor condition, were you able to get that reduced response there? But that's what we think may have been going on. And so our take-home message is that context matters. So if you think back to the zebra and the lion, well, what if that zebra is a few minutes away from dehydration and collapse? Well, then maybe it's worth it to take the risk of getting a drink next to the lion. So context matters. All right. And so finally, just to wrap up, just to tell you what I'm sort of working on in Connecticut, one of the main things I've been doing, I actually switched to a terrestrial species, the redback salamander, but I did that for a very good reason. I joined a research collaborative, we call it SparkNet, the Salamander Population Adaptation Research Collaboration Network. And what's really cool about this network is we were doing a long-term mark recapture study with redback salamanders. We're using identical methods and we have identical plots set up across 29 different locations across the Northeast, Upper Midwest, and into Canada. My plots are the only ones in Connecticut, and so we're representing there for you. And what we're able to do with these techniques is look at broad regional scale patterns of body size, of survival, of fecundity, and we're specifically interested in some of the effects of climate change. And so, for example, we have a study we're getting ready to submit to Ecology, testing the prediction that climate change, global warming, will lead to changes in salamander body size. So that's sort of what I'm doing now. That shows you all of our sites, just to give you an idea of how widespread this is. We have academic institutions, we have USGS, United States Geological Survey, we have US Fish and Wildlife Service sites. So it's a pretty fun, pretty cool project. And it's a great way to get students involved as well. So that's just a picture of some of my students helping me do surveys 1 year.
I was just going to say, how long will this effort go on?
So far, we don't have an end date set. My plots have been going since the fall of 2016. Some of the plots have been going for almost 10 years now. So we've added to the network as we could, and so we'll continue on. It's a wealth of information. You know, I've got an independent study student right now. She's able to assess, because we have so much data, the ecological differences and the different color morphs of the salamanders. There's been some work on that. I've got another student getting at some predator avoidance stuff and how that affects territoriality and site fidelity. And that has importance when thinking about climate change. As we see range shifts, species moving more northward, right? They're going to be encountering more and more predators. So stuff like that.
What's the average life of a salamander?
For the red, it depends on the species. For the redbacks, so common knowledge in the literature is that it's 3 to 5 years, but I've got individuals that I first collected as an adult that are still alive. I'm individually marking them, so I know who they are, right? That I am finding 5 years later, going strong, laying eggs, doing stuff like that. So maybe longer than we previously thought, but this is the first study of its kind with this particular species. The Eurythia nana, the ones I worked with down in Texas, those were about a 5 year expectation, a little bit longer in captivity, maybe another year. But
What do you think? Maybe you know some of the conclusions now, but what has this study told you so far?
So far, we know that salamanders are not meeting expectations as far as what we would predict with body size changes and warming climate. Um, yeah, we're just starting to dig into the analysis, right? We had to accumulate about 10 years worth before we could start getting good answers. But that's one of them. We're not finding a lot of influence of morphology. There's been some publications that have suggested, for example, that unstriped morphs handle cold temperatures better, whereas striped morphs handle warm temperatures better. Other studies say we don't find that, or they find the opposite. And so we're being able to get at that. We're not finding a lot of relationship with morphology. Our hope is we can finally put that little question to bed.
Kristen, what's known about the nature of the chemicals that are seen as these alarm signals?
Yeah, so there's been some work on those. Um, they appear to be compounds. So what prey are using to identify the predators, it's a combination of hormones, of waste products, and then just like general odors, just like our own body odor is the same thing, right? Waste products and hormones. And so there's been some work on it, though honestly, there's less interest in that. We do know that diet, the actual digestion of prey influences how a predator smells, and that's across contexts in a lot of different species.
I want to go back to one of your earlier points where you're talking about the opportunity costs and that I think you said some of the salamanders were less fit. Given the that spring waters or where all those fish were,
Mm-hmm.
will that group of salamanders, if they don't move or don't do something else, will they eventually fade away? I mean, if there's that many fish around, you can't come out very often to do other things that are going to keep you alive. Is that correct?
From what we can tell, they're mostly eating what they could find underneath the rocks and logs. And so they're just not coming out much. But so far, monitoring of the population of that particular species seems pretty stable. There's some suggestion, though we don't know for sure, that maybe they are taking further refuge down into some of the aquifer vents, but I don't know for sure. We don't know. It's they've tried to do dye trace studies throughout that region to figure out what aquifers and rivers flow where, and it's at one point you'll put some dye into a place and it won't show up again for 20 miles in a completely different area, but then another one you'll put dye in and it shows up 5 feet away in another vent. And so even geologists are not completely clear on the connections there, and cave diving is so dangerous it's been hard to map. Any other questions? Guess I should give some acknowledgments here to a lot of this work. A lot of people helped. And yeah.
all grad students that work with you?
I'm sorry?
Are these undergrad or grad students that work with you?
There were some undergrads, there were some graduate students. I was a graduate student myself doing most of this work, though I did some of it in postdoctoral work. But yeah. Yeah, like the guy standing there holding the fish, that's Ray. He was an undergrad that I mentored for a couple years. He's actually on one of my publications working with the Texas blind salamander, which is a different species from that same complex. So, but he helped me do a lot of collection. You know, it's so if you're someone who likes to go fishing, the Spring Lake, because there are 5 different federally protected species in there, not just the salamander, but also the fountain darter and a couple species of aquatic plant and things like that. It's considered protected habitat, right? And so you need special permissions to do anything in there. It's part of the reason the fish population is so bad. There's no fishing allowed. And so we collected predators using the old hook and line method. And so it's one of those rare occasions where they're like, well, got to go fishing for research, and you know you're going to catch stuff.
How did the game fish get introduced into there then?
So the dam was originally built when they converted that spring system, that headwaters region, into the first theme park west of the Mississippi. And so, just as examples, right? Let's see. The very first Tarzan, I believe, was filmed there. They were famous. They had a submarine underground theater, and they had like a swimming pig that would perform. And they also had women who dressed up as mermaids, and they would perform for the theater while you were all submerged. They had some rides.
There's so many bad jokes we could make at this.
Oh no, it's it's very 60s, right? It's where the wealthy would go, and they filmed the 80s flick Piranha there, and that's when they introduced all the elephant ear that now use more water than the entire city of San Marcos every day, right? They're nearly impossible to get rid of. It's been really neat over the last 15 years or so. So not only did that property get donated to Texas State University for use, over the last specifically 6 years, they rolled in there and they took out every man-made structure that wasn't originally there except for a boardwalk over a wetlands area. And they have done everything they could to return it to its more natural state. They converted the old hotel into a research and learning center so people can go visit that. They still do glass bottom boat rides so you can see all the fish and all the cool stuff that's in there. And the one thing they haven't done yet is remove that dam, because it's going to turn it back into the flowing swamp that it originally was. And there's a lot of pushback on that from the public, and you always have to get public input for stuff like that. So that's the one thing they haven't done.
What, other than somebody that wanted to develop it, why would there be pushback on turning that back into a flowing swamp?
Because then you lose the lake aspect, and the visitors like the lake aspect. They like to walk along the lake. They like to take the glass bottom boat rides. Yeah, they say the theme park is part of the history too, and so yeah.
Do we have any other questions? Well, thank you very much. This has been a really interesting talk.
Yeah, I hope it wasn't too nerdy for you.
It was very interesting.
Yeah.
To be honest with you, I didn't know much about it, and that's what's great about these clear sessions, because you find out stuff that you never thought you'd even think about. So thank you very
Yeah.
much.
Yeah, no
Yeah.
problem. I thought about doing some of the redback stuff that we have so far, and I was like, let's do something they're less likely to have heard about.
And we learn new terminology, like new ways to use the word plastic.
Yeah, no, it's
And your credit card, right, Pat?
yeah.
Thank you, Kristen.
All right, you're welcome. Y'all have a good day.
Yeah, thank you. You too.